Hello, my name is Caroline Ennis, and through the Creative Inquiry Program, I've been a member of the Bairdii Lab since 2020, studying mitochondrial genomics. Today, I will present my work to annotate and analyze the complete mitochondrial genome of the forest dweller, Baird's tapir. First, I will introduce some background information about this project in Tapirus bairdii, followed by a brief explanation of the methods used, an analysis of my results, and finally, the future directions of this work. Mitochondria have a small, circular, double-stranded genome. Studying the mitochondrial genome offers significant advantages to nuclear genomes. They're easier to isolate and can even be extracted from environmental samples. Tapirus bairdii is found in Central America, ranging from Mexico to Colombia, grazing in the forests and the wetlands. They play important ecological roles, but are threatened by many anthropogenic activities. In 2014, they were classified as endangered by the IUCN. There are some conservation efforts in effect, as well as some successes with captive breeding. Despite their endangered status, however, very few genomic resources exist for the genus Tapirus. One study in 2004 indicated Baird's tapir, in particular, was at an increased risk for loss of genetic diversity due to inbreeding caused by population decline. Mitochondrial genome data is a step towards indirect surveillance, which is a preferable method for population sampling for elusive populations like Baird's tapir. Looking at a tapir's short trunk, you might think they would be in the same order as elephants or even anteaters, but actually, they're a part of Perissodactyla, the order of odd-toed ungulates. This order includes tapirs, rhinos, and horses. They are one of 4 extant species of Tapirus, which vary in coat color and geographic distribution. There have been contradictory studies concerning the phylogenetic relationship between the species within Tapirus, and even a 5th species has been proposed by more recent data. Therefore, there is a need for additional genetic analyses to resolve this issue and provide evidence for the exact order of speciation. The objectives of this study were to first annotate and characterize in detail the mitochondrial genome of Tapirus bairdii. Secondly, this new mitochondrial genome was used to explore the phylogenetic position of this species within the genus Tapirus based on the protein coding genes of the mitochondrial DNA. The ultimate goal is to use this new information to support conservation strategies focusing on Tapirus bairdii. Our data will help in resolving taxonomical issues within the genus and will serve as a base for environmental bioprospecting and biomonitoring of this species. So, how do we do this? Collection, extraction, Illumina sequencing, and assembly were conducted from a specimen in Mexico. These steps permitted the assembly of a full mitochondrial genome that was then analyzed in detail. The field of genomics has progressed rapidly in just a few decades to where a file of 16,000 nucleotides can be turned into discernible figures and useful data using bioinformatics. This is an overview of some of the tools used in this process. Each of these tools analyzes the genome or sections of it to provide different information. Mitos gives a rough annotation of gene location, which is then manually corrected by analyzing the reading frames of each gene using XPOSI and MegaX. Forna and RNA structure servers predict the secondary structures of the tRNAs and control regions, respectively, based on the lowest free energy. CGView allowed me to create a circular map of the major features identified. Mitofast and IQ-tree softwares were employed to infer a phylogenetic tree using the protein coding genes from the species across Perissodactyla. Given the success of these tools, what is the purpose of biologists after inputting the data? Well, these tools are not perfect and cannot troubleshoot the changes and abnormalities of biological systems like gene overlap, so manual correction is needed to refine computer predictions. As is standard in mitochondrial genome studies, a visual map shows the gene locations, directions, and types. The 16,697 base pair genome of Tapirus bairdii contains the expected makeup of 22 transfer RNAs, 2 ribosomal RNA genes, and 13 protein coding genes. The origins of replication for both the heavy and light strands were identified, and a majority of the protein coding genes were located on the heavy strand, as expected. The sizes of the genes and other genomic components were all within the expected range or identical to related species. The gene order is identical amongst closely related species, supporting the observed stability of mitochondrial genome arrangements in mammals. Nucleotide bias is the comparison of the percentage of each nucleotide's frequency in a given sequence. On a genome-wide and gene level, there was an AT bias. Characteristic of mitochondria, the genome is compact with small intergenic spaces and some overlapping genes. The protein coding gene codon usage was predicted using the codon usage web server. The typical vertebrate mitochondrial start and stop codons were present, with no non-canonical initiation codons found. Some of the protein coding genes terminated with a T, indicating a polyadenylation mechanism for termination. The same genes, NAD2, COX3, NAD3, and NAD4, also had reported truncated stop codons in other members of Parasidactyla. Selective pressure analysis compared the non-synonymous substitution rate to the synonymous substitution rate for each of the protein coding genes. All of them did not surpass the value of 1, indicating that the same genes experienced purifying selection. ATP8 showed a much weaker constraint compared to the other PCGs. Selective pressure analysis in other species of Tapirus has not been done and is generally poorly studied in Parasidactyla. There was one previous study in equus, or horses, that also indicated overall purifying selection in mitochondrial protein-coding genes. The secondary structures of the tRNAs exhibited the typical cloverleaf structure, except tRNS1, which was observed to have a missing D arm, common to almost all metazoans. The anticodon pattern showed similarities to other members of Parasidactyla and were nearly all identical to the closely related Asian tapir. The control region contains stem loop structures, which are found to be under selective pressure in mammals. This region is also highly repetitive, with 22 microsatellite repeats and a long tandem repeat sequence similar to other members of the order. A high degree of conservation was observed in the central domain between Baird's tapir and other members of Parasidactyla, a common occurrence in vertebrates. A multiple alignment of the control regions helped identify the functional domain regions and conserve blocks. The extended terminal association sequences block and conserved sequence block had higher nucleotide variability due to higher substitution rates compared to the central domain. Control regions are often used in population studies for mitochondrial genomes of mammals, so this was an important feature to explore. There had yet to be a mitogenomic phylogeny created using Tapirus species. This analysis confirmed the monophyly of Parasidactyla. It also suggested T. bairdii and T. terrestris were the most closely related within the genus. As previously mentioned, mitochondrial genomes are useful for population genetic studies in threatened species. Comparative studies of mitochondrial DNA can signal population changes and, in conjunction with nuclear DNA, contribute to long and short-term planning. One example of this is extrapolating haplotypes from mtDNA samples and monitoring maintenance of genetic diversity, like this study of the Chinese water deer, a small mammal under similar pressures to Baird's tapir. This can be used to make recommendations and decisions about transplantation and captive breeding programs. Understanding current trends in mitochondrial genome diversity allows detection of genetic changes given the rate of evolution is different for different parts of the mitogenome. Creating genomic resources assists in the understanding of selective pressures and evolutionary relationships, critical data for threatened species. This genome can act as a reference for marker retrieval in bioprospecting and biomonitoring. Since there are multiple copies per cell, mtDNA is easier to isolate from less tissue or environmental samples. Indirect surveillance techniques using environmental mtDNA are currently being explored for larger mammals, and this genome will assist in future efforts with Baird's tapir. I'd like to thank Clemson Creative Inquiry, the Department of Biological Sciences, and the Honors College for their support of this project. For more information, please see our recently published paper, and thank you for watching.